Today we are going to discuss about dentin and the process of its formation, and it's called as dentinogenesis, and the cells that are involved in dentinogenesis are your odontoblasts. Before we dig in deeper, first let us run the intro. Dentinogenesis is a continuous process that happens throughout one's life. It only stops if the pulp tissue is removed. This is because the cells that produce the dentin, odontoblasts, are so programmed that they even produce upon any kind of external stimuli. For example, the stimuli can be in the form of dental caries or dental trauma. Because of that stimulus, the odontoblasts are programmed to produce a specific type of dentin, and that is called as tertiary dentin. Now, based on the time of its production, dentin can be three types. If any dentin that produces before the root formation is complete in a tooth is your primary dentin. Similarly, any dentin that produces after the root formation is completed, and that is your secondary dentin, and the tertiary I have already mentioned. This is exclusively based on the time of its production. It can be any kind of dentin, but where are these odontoblasts coming from? To understand this, we need to go back to the Bell stage of odontogenesis, where one of the main events that occurs during your Bell stage is your crown pattern determination, where the shape of the tooth is actually getting determined. Once the cuspal pattern of the tooth is decided, there is a proper interaction happening between inner enamel epithelial cells located within the enamel organ and the cells that are located within the ectomesenchyme. Before we actually see the interaction happening between these two cell layers, first let us look at cells that are located within the dental papilla, and these are the ones which are going to be differentiated into odontoblasts. These dental papilla cells are actually derived from neural crest cells, and these are distributed evenly all over the dental papilla. Now, the cells which are located at the periphery of the dental papilla are closer to the inner enamel epithelial cells, and these cells are continuously dividing. Once the cell gets divided, the daughter cells has two options. One, the cell can either remain within the periphery of the dental papilla, or the cell can decide to come to the pulp core. The cell that decides to remain within the periphery of the dental papilla has the high chance of getting differentiated into odontoblasts. Other ones will start moving towards the pulp core. During the crown pattern determination, when the interaction is happening between the inner enamel epithelial cells and the ectomesenchymal cells, the inner enamel epithelial cells start producing some signaling molecules, like for example transforming growth factor beta, and these kind of signaling molecules pass through the basement membrane, which are actually consumed or taken up by your peripheral located dental papilla cells, and these signals act as differentiating factors, and the cells starts differentiating into your odontoblasts. Now, a differentiated odontoblast morphologically differs from an undifferentiated cell within the dental papilla. An odontoblast is now elongated, columnar, it has a long cell body and multiple cellular processes. If you notice, I use the word multiple cellular processes. This is only to begin with. As the differentiation process continues, these cellular processes will unite to form one significant single cellular process, and that is called as Tome's fiber. Remember, it's not Tome's process, what you see in ameloblasts. These are called as Tome's fiber. It is well documented in the literature that the cell body of a differentiated odontoblast will secrete the collagenous matrix. Similarly, the cytoplasmic process, what I just mentioned as Tome's fiber, will secrete the non-collagenous proteins. Now, dentin, like any other hard tissue except your enamel, has two basic components within its organic matrix. That is your collagenous proteins, chiefly consisting of collagen type 1, 3 and 5, along with non-collagenous proteins like dentin sialoprotein, dentin phosphoprotein. So collagenous proteins and non-collagenous proteins together constitute within the ground substance that makes your organic matrix. And this non-mineralized or unmineralized organic matrix that is produced from a differentiated odontoblasts is called as predentin. Because of the fact that the dentin production occurs throughout one's life, it is accepted that at any given point of time, one should be able to see some amount of predentin, because there is always a time lag that occurs before this predentin actually gets mineralized. Now, when we speak about mineralization, the way the dentin mineralization occurs is totally different. Now, to understand this, we need to talk about the types of dentin, not based on the time of its formation. Instead, we need to discuss about the location of dentin. There are different types of dentin based on location within the tooth. The dentin that forms just beneath the dentinoenamel junction, or the dentin that forms first, is your mantle dentin, and the remaining majority of the dentin that forms below the mantle dentin is your circumpulpal dentin. It's called circumpulpal because it's going to encircle the whole pulp all around this tooth. Now, the way the mantle dentin gets mineralized and the way circumpulpal dentin gets mineralized is totally different. To understand this, the mantle dentin mineralizes through a mechanism called as matrix vesicle, that is your first form dentin. Likewise, the circumpulpal dentin mineralizes through another mechanism called as heterogeneous nucleation. To understand the matrix vesicle theory and the heterogeneous nucleation, please check out my other video, which the link of which might pop up somewhere here. Briefly, to understand the mineralization process involved in dentin, this particular organic matrix that is produced by the differentiated odontoblast can get mineralized either through two mechanisms. One, the globular way or the linear way. So during the heterogeneous nucleation, where the nucleating sites that actually attracts the calcium and phosphate, so in the dentin, during such attraction, multiple calcium and phosphate ions come together. They form the calcification sites or globular calcification sites. These globular calcification sites are called as calcospheroids. To understand better, let us discuss in this way. Whenever there is rapid dentin production, there is less time for these calcospheroids to fuse homogeneously, and because of this, there are defects happening between the globules. In other words, when the dentin production is slower, like in your secondary dentin deposition, during secondary dentin, the rate of dentin formation is slower. So what happens, there is ample time or sufficient time for these globules to fuse homogeneously, turning this globular way into linear way of calcification. During rapid dentin production, the globular way of mineralization predominates, and similarly during slower dentin production, the linear way of mineralization predominates. This can be especially seen especially at the junction of the mantle dentin and the circumpulpal dentin, wherein which lot of defective unmineralized areas can be seen histologically, and these unmineralized areas are called as interglobular dentin. It's just the spaces that remains unmineralized within those globules, and this interglobular dentin is actually a defect in the mineralization process and not in the organic matrix production. This is seen, actually, if we see in the histological section, we can see the dentinal tubules passing through the interglobular dentin, suggesting that the mineralization has not occurred and not the organic matrix production. I hope this video has helped you understand dentin and the process of its formation. If you have liked this, please do not forget to hit that like button, and please share with your friends. And please be generous and subscribe and support this channel. Looking forward for all your support. Thank you. See you in the next video.